Thank you for coming tonight. This is our third lecture this year for the Science and Engineering Lecture Series here at AU. Tonight we have one of our new faculty, Dr. Kyung Shin Kang, is from, well, he did his undergraduate and graduate work in Korea. But we found him as a postdoc at the IU School of Medicine, where he was doing some biomedical engineering. So he's come to us. He's in that first year faculty battle of trying to keep his head above water, but he's doing a really nice job for us, and we're really glad he's here. He and his lovely wife have a young son, Ian, who just turned 3, and he's starting to talk and get himself into a lot of trouble, I imagine, at home. So tonight, let's welcome Dr. Kyung Shin Kang.
Thank you. As you introduced, my name is Kyung Shin Kang, and Physical Sciences and Engineering new faculty. Thank you all for coming today and listening my talk. I want to start just warning you, since English is my not native speaking language, so if you don't understand, help each other, help neighbors, okay? And then still you don't understand, don't hesitate to interrupt me, so that I can explain, rephrase one more time, okay? I started my professional career here as an assistant professor last year. Yeah, and I don't see Dr. Willis Troy here, but I rely on Dr. Troy for a little bit of everything. The other day, we talked about retirement. Yeah, seriously, I just hired, and we're interested in retirement. There are many reasons for that, and not only financial aspect, but also we know that we're going to live longer than my parents' generation. That's because I think thanks to the advance in engineering technology to help and heal all the issues in our body. So I want to talk about how engineering technologies are contributing medical applications and how it contributing to increasing your lifespan. So sometimes life is not easy. Bad things happen to you, right? This is an example dislocating one or more of your body part. Or you may have job-related injuries or wound, or you may have serious disease, so that you may have to replace one of your organs or tissues, right? This is one of typical examples that restore the function of lower limbs and others. This is Carl Brasher, the first African American to become US Navy master diver. I don't know how many are aware of this man, but the story was filmed in 2000 at the Man of Honor. And he became master diver despite having a left amputated leg. Isn't it amazing? This is pretty inspiring to me. And a lot of people are dreaming of having robotic arms and robotic eyes to have better life, right? So probably this shows a future of our biomedical applications of engineering. And this man have left bionic eye, right arm, and both legs are replaced by robotic legs, so that he can run more than 60 miles per hour. Let's take a look at current status of technologies each. This is Hugh Hare. Am I pronouncing right? Hugh Hare? Yeah, Hare at biomechatronics group at MIT. He was one of top rock climbers at the age of 17, but because of severe frostbite, he lost both legs below the knee. So he had to develop his own prosthetics to overcome the limitation, so that so this is his own invention. So now he is able to extend his legs. Also, he can climb ice very easily. So let's take a look at his interview.
My research group here at MIT has a long history of innovation. We developed an artificial knee to be used by people that are amputated above the knee. That has a brain, it's able to sense its environment, so it auto-adapts. As a person moves, it figures out how to modulate its own resistance to enhance the biomechanics of its wearer. We've developed a bionic foot-ankle device. This is the first prosthesis in history that's powered, that emulates lost muscle function. With this, it actually injects energy into the walking or running gait. Within minutes, often, a person can walk normally and says to us, I have my body back, I have my life back. Here at MIT, we're advancing the fundamental interfaces between the human body and the built, designed world. Electrical interfaces, mechanical, dynamic. We are developing the fundamental building blocks for the elimination of human disability. So I just love designing bionics. I love every few months upgrading my own legs so I have better balance, I can move faster. It's interesting because my biological body as I age gets worse and worse due to age-related degeneration, but the artificial part of my body gets better in time. So I predict when I'm 80 years old, my balance will be better than an 18-year-old.
Did you just hear that? His biological body is degenerating while his amputated robotic legs are updating. Isn't it amazing? Yeah. By the way, how much do you think it cost per leg? 10,000. Actually, roughly $50,000 per leg. Okay. Still, this is cheapest one among leg, arm, and eyes. So this is an example of bionic prosthetic arms. There are multiple cases that received this kind of robotic arms. Actually, the amputation site can send some sort of neural signal to the robotic arms. So these guys have a lot of sensors on their amputation site, and then the intention from the brain can be transmitted to the robotic arm so that a robotic arm can interpret it into a motion. So both arms are, I don't know how many you guys have seen the Baxter robot in engineering center, but that robot has 6 degrees of freedom per each arm. That means 7 different joints each arm. Each arm over there has more than 30 degrees of freedom, means 30 different actuators mimicking the arm motion. Does it make sense? It's more complicated structure. How much do you think it costs? 35? 40. I want higher. $500,000 actually. Each arm. So this is last one, the eye. Take a look at the eyeball here. Do not take a look at this eyeball, the electrode array over there. The eyeball is just support. This man has macular degeneration leading to total loss of his central vision. So from the glasses he's wearing, there is a camera. So camera can capture some sort of image, and then here is a transmitter so that the image from the camera can be transmitted wirelessly to the electrode array that was implanted on his retina. It doesn't give a full vision like we see. However, using this technology, he now is available to see, recognize just a blood shape of the door, window, so that he can walk without any assistant now. It costs $150,000. What about internal organs? Can you recognize this picture? As a punishment, yeah, Prometheus. As a punishment for giving humanity the secret of fire, he, the Prometheus of Greek mythology, was chained to a rock where an eagle would eat his liver here each day, and every night his liver regenerates itself, so that starting the torturous cycle over and over. Does it make sense? So this may sound like ancient version of a horrible movie, but actually it's interestingly consistent with actual human physiology. That means your liver can regenerate itself. Even though you are alcohol abused or medicine abused, your liver can regenerate itself. Isn't it interesting? And also, that means you can donate up to 60% of your liver to other people. So over time, your liver will be back to the original size. So this is from statics. Take a look at this gap between the number of waiting lists and then number of donors. See, over time, the gap is growing up gradually. And now, 116,000 people are waiting, and 20 people are dying each day waiting for the organ donation. And only 3 in 1,000 people die in a way that allows for organ donation. So, without any other help, it's going to be more severe than before. Then, why do we have to wait for the organ donation? What is the reason? Any idea? Why do we have to wait for organ donation if we lose some sort of function of one of our organs?
Not enough organs.
Because organs do not grow on trees. So it's not possible. You cannot order your internal organ whenever you need from Amazon, right? Or some people can say that we are different from axolotl, who or that have mastered the regenerating skills to perfection. So even though it loses one of its arm, it can restore it back. But we are not. We don't have that kind of ability, right? So certainly, scientists, engineers, and medical doctors have been trying to close the gap between the waiting list, number of waiting lists, and the actual transplant number. And one of the technology is that making artificial organs. This is an example about lung, artificial lung. So as our lung does, this device mimics the changing carbon dioxide from your blood for the fresh oxygen from the air. The problem is that this is huge. This is the device. So it's not implantable. It's not ready yet. It's too big. But someday in the future, it may be possible because the ENIAC, the first computer developed in 1940s at UPenn, this was room-sized. Now you carry a smartphone much faster than this ENIAC. So someday, we are going to have really small artificial lung. This is an example of world's only FDA-approved artificial heart device. So when it first came out, 2002, maybe 4, the driving unit was 400 pounds. So it's not possible to move around with that driving unit, right? But now, the second version is this size. So it's getting better. But still, there is a gap between the actual organ and then artificial organ. Maybe that's why people are thinking of having their own clone. So whenever they want a certain body part or internal organ, they can harvest from their own clone, right? Does it sound horrible? Maybe a human clone is not legal or ethical, but there are some researches are going on about the possibilities using animal as a test bed or a platform or some sort of clone. So this is an example. In 1997, we call this Vacanti mouse because the doctors and brothers Vacanti at Harvard University developed this model. So they fabricated some sort of ear-shaped mold with biodegradable material, and then they seeded cow-derived chondrocytes on top of that scaffold, and then implant it on the back of the mouse. So this is the most first trial for tissue engineering. And for the internal organ case, now we have technology to grow fetal kidney in rat or mouse or those animals. So I want to introduce the concept of tissue engineering. By the definition by the Langer doctors, Langer and Vacanti, it says interdisciplinary field that applies the principles of engineering and the life sciences toward the development of biological substitutes that restore, maintain, or improve function or whole organ. And there are many different definitions, but the concept is this is an engineering, as the name implies, tissue engineering. Actually, this is multidisciplinary field, combining all the mechanical, electrical, material engineering sciences, and then medical field. A lot of different areas are combined in this field. So basic concept is on your right. So to avoid the immune rejection response in your body when it is implanted, it's critical to start using your own cells. And then expand it until you have enough number of cells. And then you may add biomolecules and cells on 3D structures we call scaffold, and then grow for some time to mature, and then implant it in your body. That's a basic concept of tissue engineering. There are 4 different categories to describe engineering complexity. It starts with flat tissue structures here, such as cornea, and tubular structures, trachea, and hollow viscous structures, bladder, in solid organs like kidney. It sounds like it's very hard to engineer these solid organs and then the flat tissue structures, but believe me, these are all same. All organs are very difficult to regenerate. The reason is that, for example, the cornea, it seems pretty simple structure, right? Just cells, some sort of blood vessels, and simple cornea structures. But nobody ever engineered perfect cornea, ever. So the flat tissue structures is not easy. To my knowledge, this is the first engineered internal organ, bladder, at Wake Forest University. So what they did was fabricating bladder-shaped biodegradable structures, and then patients' own cells, they seeded them on top of this bladder-shaped scaffold. And after growing over time, they implanted into 7 different patients, and the result was pretty good. there was no mucus production. Mucus production was normal, and then urinary calculi was sort of okay, but almost no forming. And the renal function was preserved, and then biopsy shows that the structural architecture was really good, and phenotypes was almost perfect to the original bladder. This is another example of trachea. In Spain, 2008, this surgeon tried to engineer patient-derived tracheas. So let's take a look at this movie clip showing how they perform to replace the trachea. So this lady had some sort of clogged airway, so that left lung was collapsed. So the only way to replace it is to replace the trachea to restore the lung function. So from the cadaver, they took out the whole trachea part, and then they removed all the dead cells because they can intrigue the immune rejection response. And then they took out the bone marrow stromal cells and epithelial cells and chondrocytes and seated on top of the stripped out trachea construct. And then in a bioreactor, they cultured it, and then over time, they shaped to fit into the patient. And then finally, it was implanted into the defect site. Unfortunately, the result was not good, even though it was published in a very famous professional journal, Lancet. The result was failed because of the side effect of some sort of implanted trachea. But that surgeon had performed same procedure into 17 different patients. And this boy is one of them, and luckily he got no immune rejection, no any other side effect, so it seems perfect to him. So the tissue engineering technology is not perfect yet. So it's case by case, patient to patient, so we should be careful to using those technologies yet. Let's dive into tissue engineering for more details. Actually, the tissue engineering relies on three different factors: cells, signals, and scaffold. Cells are basic building unit, right? Cells can secrete extracellular matrices, so eventually they can build new organ over time. The scaffold is 3D structure because our body is three-dimensional shape, not cells in a petri dish, right? They need some sort of microenvironment. So we have to provide some sort of scaffold. And then signals can order cells, do this, do that, go this way, go that way, something like that. So this can be chemical signals or physical signals. So let's take a look at cell briefly. So as I mentioned earlier, to build your own organ, it's critical to use your own cells. So the first step starts with harvesting your own cells, depending on the internal organ that you need. And a lot of different cells can be used, precursors. And like for bone generation, you can use bone-forming osteoblasts, or you can use stem cells. Stem cells have a very unique property like self-renewal, which is great, and it can be differentiated into so many different lineages. This is an example of adult mesenchymal stem cells. It can be differentiated like fat tissue cells and bone tissue cells, cartilage cells, neuron cells, and muscle cells. But the problem is we don't have full control on the differentiation lineage yet. So it is promising, however, it's still risky to use. So let's take a look at the signal part. As I said, the signals, traditional signals can be just biomolecules, such as growth factors. However, nowadays, many people found that microenvironment surrounding the cells and then physical stimuli are also important. So this is a finding from one of mechanical engineers at University of Pennsylvania. He found that the matrix stiffness can potentially regulate cellular activities. Does it make sense? So let's take a look at this. So he prepared 3 different types of matrix stiffness. One, soft, which is similar to brain. And second one is intermediate soft matrix, which is similar to our muscle. And third one is hard matrix, which is similar to bone. And he seeded same type of human mesenchymal stem cells. And then he observed different morphology. So in soft tissue and intermediate soft matrix and hard matrix, without any chemical treatment, how rigid it is can induce different differentiation. Does it make sense to you? So take a look at this picture. So tubulin is brain-specific marker. So when the mesenchymal stem cell turned into neuron cells, they express tubulin. Take a look at that. So only soft matrix, only cells grown on soft matrix expresses tubulin, which means they turned into neuron cells, but not from intermediate and hard matrices. MyoD is muscle-specific marker that is expressed only intermediate matrix stiffness case, and bone-specific markers was expressed only hard matrix. So that means the cell's microenvironment is really critical to their cellular activities. So these findings was led to a lot of different areas, including computational analysis. So, for example, all cells are exposed to different types of physical stimuli. For example, we have blood circulation all the time, right? So naturally, the internal surface of our blood vessels are exposed to continuous shear stress. Also, as I walk like this, my lower limbs are exposed to cyclic compression and tension, right? That means my bone cells are experiencing compression and tension continuously as I walk. My lumbar spine experiences continuous compression because of my body weight. So using this computational simulation, we can mimic or we can predict first how much magnitude our cells are experiencing those mechanical stimuli, and then we can recreate the same environment in vitro. So these are examples of imaging technologies. So thanks to the advance in imaging technologies, we can have now very accurate MRI and CT data, right? So from those informations, we can personalize those simulation results. And also, we can use that information when we design 3D structures. I don't know how many of you can recognize these 2 companies, but these 2 companies are famous for selling traditional universal testing machine. Means they usually sell testing machines, but nowadays they use their skills into different area, so that they now sell bioreactors, so that researchers can grow patient-specific constructs in their bioreactors. not only compression, tension, and shear stress that does exist our body system, but also extra like non-biomimetic stimuli such as ultrasound and electromagnetic field can be utilized to manipulate cells activity. This is an example about electromagnetic field. So this group uses adult mesenchymal stem cells and then stimulate with electromagnetic field, time-varying electromagnetic field. If you take a look at these two graphs, you can notice that depending on the frequency, the bone-specific markers expression is different. And this RUNX2 is another bone-specific marker, early-stage marker. And depending on the frequency, they have now different expressions. That means without any chemical treatment, the non-biomimetic stimuli still can regulate cellular activities so that we can treat patients even outside the body. Some people say that we're not God. We can create exactly our body does. So some people try to use our body system as a bioreactor. We call that in vivo bioreactor. So instead of using in vitro bioreactor that I just introduced, they use patient's own body to regenerate the tissue. For example, let's say I have a defect on my tibia here, and I need my own bone. And the doctor is trying to grow my part of tibia into on my hip or spine somewhere else. Does it make sense? So this is an example for bone regeneration. They try to make some space between the periosteum, which is a thin layer on your bone, and the tibia bone here. So they create some sort of space in between them, and then they injected hydrogel to maintain that space, and then close the periosteum here, and then let it grow. Over time, they have new bone because the periosteum is full of fibroblasts and stem cells, so that those host body cells can migrate into the hydrogel, and then they form new bone. So this is histology. So this was original bone, original cortical bone of tibia, and this is bone marrow part. And this means inside. This is outside. So if you take a look at that part called bioreactor, you can see cortical, lamella, woven bones, which is typical type of bones. So eventually, they were able to take this new bone here, implant it into the defect site, and observed a great result. So last factor is scaffold. So this is an old way to fabricate 3D porous scaffold. So after mixing salt and polymer, after solidifying, you can immerse it in the water so that salt can dissolve in the water, right? That means the place where the salt was, now it becomes empty space, means pores. So this scanning electron microscopic image shows that where the salt was, and now it became the pore. The problem is that these pores, pore size is not that much controllable, and also a lot of pores are not well connected each other. So cells cannot grow into very well. And thanks to the advance in 3D printing, Now we are able to fabricate patient-specific and well interconnected 3D structures. So that movie shows basic layer by layer process of 3D printing. So as you know, dot becomes line, line becomes area, and area becomes finally volume, right? So eventually we are able to fabricate 3D structures like that. The beauty is that from the imagings like MRI and CT, we can have patient-specific structures like this. So this is an example about 3D printing based scaffold. And one of the advantages is that we can now incorporate growth factors. This BMP stands for bone morphogenetic protein 2, which is critical for bone regeneration. And this can be incorporated into 3D structures in microspheres here, so that the scaffold can release sustainability. They can release BMP2 over time, as the graph shows. And this is typical example of how we test bone regeneration using rat or mouse models. So we create a hole here on the cranial side, and then if the defect size is greater than certain limit, for example, rat, if rat has a defect 8 millimeter in diameter, then the bone regeneration cannot be observed over time. So if we don't put anything, then of course there's no bone regeneration. This is salt-leaching scaffold, and just 3D printed scaffold, and BMP2 releasing 3D printed scaffold. Of course, the last one shows the faster bone regeneration. The reason we have bones on the parameter is that they didn't see the cells on the scaffold. So that means the host cells migrated into the defect site from the neighboring bone and formed new bones. So thanks to the 3D printing technology, now people can fabricate all parts of human body. So ears, nose, and skull, and limbs. But don't be fooled. This is not fully functional yet. This is just structures. Here's another example of a skin printer at Wake Forest University. I'll say this is more like a moving spray. And on the wound site, they use biomolecules and biomaterials and cells all together and then spray over the defect site after scanning the defect depth and size, overall size. And they claim that this method is 3 times faster than traditional skin graft. And this was funded by Department of Defense, so it was a pretty big project. But as I said, it's a very, very early stage of prototype. This man is Anthony Tala, the director of Wake Forest Institute for Regenerative Medicine. And in 2011, he showed 3D printed bladder in TED Talk. But this was not fully functional. This is the guy who made the first bladder in the world. And he engineered more than 15 different organs at Wake Forest University. But as it mentioned, this is really, really early stage of tissue engineering. So even though you see bladder-like structures, don't believe that it's fully functional. So another people claim that no matter how good quality we have for the biomaterials and growth factors and cells, we are not able to mimic the nature of our organ structures. So some people have been trying to use the own tissues. So this is an example of rat heart. They, in order to transplant one rat heart into another rat, they decellularize all the cells from the donor's heart. So as you can see over time, the color is changing to white because there's no blood, no cells. The histology shows almost nothing. And this is the heart-specific marker expressing in the cell. As you can see, these two are before decellularization, and these two are showing the after decellularization. So the blue dots are DAPI that you can stain on your on cells nuclei. If there is no DAPI, means that there's no cells nuclei, means the cells are stripped off. And they seeded the patient's own cells. Can you see that? It's beating. That beating is just nature of heart cells. So even though it's beating, it looks like beating, it's not fully functional yet. So decellularization is more like this, the stripping of all cells from the donor's the organ. The reason we do this is that liver has its own structure, heart has its own structure, and bone has its own structure, right? So since it is very hard to mimic those structural architecture, we just want to use donors' own structures. These cells are main source to induce immunorejection in your host body. So if we get rid of all the cells, then there must be, ideally, no immunorejection. So this work has been done in Wake Forest University. This is liver. This is before decellularization. You can see it's red. And moderate treatment, and after getting rid of all the blood, it looks less red. After all the entire process, it looks white now. And you can see through the fluorescence microscope, all the vascularized networks are intact. And they seeded the patient's own cells on the structure, and then they saw the hepatic unique structures. The liver must be hexagonal structure. And the patient-derived cells started forming the hexagonal structures like that. And now, those decellularized materials are printable. So from cartilage tissue, heart tissue, adipose tissue, and we can delete or get rid of all the cells. As you can see, there is no DNAs at all, meaning there is no cells. And you can make it as a hydrogel material, and then you can print it as a structure. The reason people use this kind of polymer-based frame is that this type of hydrogel material is not rigid enough so that they cannot maintain their own 3D structures. So still, we need we have a lot of things to overcome technically. And a company in Japan is trying to make scaffold-free structures only from the extracellular matrix from the cells. So starting from obtaining sphere-like cell aggregates like this, the robot arm can manipulate those aggregates into the needle array here, so that eventually they form 3D structures. And since they are individual aggregates, they are not connected each other yet. So over time, those cells secrete their own extracellular matrix, and eventually they will combine together. So eventually they were able to get these 3D structures without using any foreign materials. And currently, tissue engineering plays a significant role. However, it is a very small role in patient treatment. So far, the bladder and skin graft and some of cartilage, trachea have been implanted in patient, and some of were successful, some of not. And more complex tissues like heart, lung, kidney were recreated in the lab setting successfully. However, we are away from being more fully reproducible, those complex tissues. However, these kind of tissue structures can be quite useful in research, especially drug development and drug screening. So, Organovo is one of pioneering 3D bioprinting company. Now, they are offering testing solution platform, like giving some sort of small tissue construct so that researchers can test their own drugs or toxicity from the food. I have another example, organ on a chip.
The lung on a chip is a microdevice that has the potential to replace animals for the testing of drugs and toxins. The device, which is lined by human cells, mimics the mechanical and chemical functions of a living, breathing lung. With every breath, air enters the lungs, filling and expanding microscopic air sacs. Oxygen is transferred across lung and capillary cells and into the bloodstream. This is also where aerosol drugs are absorbed and where infections and tumors form in the lung. The lung on a chip is crystal clear, flexible, and about the size of a small computer memory stick. But it contains tiny hollow channels created using microchip fabrication techniques. A porous, flexible membrane separates the two channels at the center of the device. The opposite sides of the membrane are lined by human lung and capillary blood vessel cells. This mimics the arrangement of lung and blood vessel cells in the air sac of the lung. Application of cyclic suction in side channels makes the entire flexible sheet and cells stretch and relax rhythmically, just like our lung cells do when we breathe. In the lung on a chip device, air flows over the top of the human lung cells, and a liquid medium containing human white blood cells flows below the capillary cell layer. To test how well the lung on a chip device replicates the natural responses of living lungs, we introduced bacteria into the air channel to mimic an infection. And we introduced white blood cells to the blood channel. We then saw the white blood cells migrate across the capillary cell layer, through the pores of the central membrane, and into the airspace where they engulfed the bacteria. Here's a video that shows this response in real time viewed through the device. The tissue cells are not visible here, but we can see white blood cells flowing freely in the capillary channel of the device, just as they do in blood vessels of a healthy person. But when we infect the air channel by adding bacteria, the immune cells abruptly stick to the surfaces of the capillary cells on the opposite side of the membrane, located directly below the infection site. Here's a magnified view showing a migrating white blood cell making its way through the first capillary cell layer, wriggling through the pentagonal hole in the flexible membrane, and then moving out of focus to the other side. When viewed from the air channel, with all cells visible, you can see a round white blood cell popping up from below. Just like in a real lung infection, the white blood cells, which are now colored red, engulf and kill the green bacterial invaders. The lung on a chip can also mimic the toxic effects of airborne nanoparticles on the lung. In fact, experiments with the device led to the prediction that breathing increases their absorption into the bloodstream, and this has been confirmed in animal studies. Bio-inspired microdevices that can mimic whole human organ function, such as the lung on a chip, could potentially replace animal testing and bring new therapies to patients faster and at lower cost in the future.
So this kind of device has a lot of different names. You can put your organ's name in front of on a chip. So this was lung on a chip. You can make heart on a chip, bone on a chip, brain on a chip. So far, we have more than 15 different organs on a chip in the world, including eye, placenta, and uterus, which mimicking women's period of 28 days. So it also has different names, like some people call it human on a chip, patient on a chip, or a lot of different names, or human emulation system, something like that. And this was possible thanks to the microfabrication technology, just like a semiconductor production. So this is basically layer by layer bonding fabrication. And now people are trying to integrate multiple organ chips in one single system so that we can test toxicity in the food, cosmetic, and dietary supplements or something like that. And this can be personalized because it uses patient's own cells. That means also we can create, if let's say you have brain tumor, and I was told that a lot of different combinations of medication are out there for brain tumor, and each patients have different responses from the medication. So if you can create your own tumor model, several of them, you can test different drug compositions, right, before treating yourself. So since this is a very promising research area, major government funding agencies such as NIH and FDA are playing a leading role in this research area. For example, last year, FDA announced that multi-year research and development agreement with a company called Emulate here, which is a startup company at Harvard University where the lung chip was developed. The goal is that to integrate more than 10 individual organ on chips in a single system. So with that, let me summarize my talk. So there are certain situations where you need to replace your own tissues and organs. And thanks to the robot arms, now you can have better mobility. And thanks to the computational simulation techniques, now we can predict patients' own situations in the body, and it can be recreated in vitro. Also, imaging technologies enabled us to mimic patients' own structures. And with microfabrication technologies, now we can personalize medicines. And lastly, 3D bioprinters can eventually enable us to have our own structures depending on the size and site of your defect. That's it.